Alex started in academia, although now he is moved to industry and entrepreneurship. But he first started his journey and as a BSc in Cell and Developmental Biology in University of Rochester, and then for his PhD, he moved to Harvard, where his main field was in chemical biology in James Bradner lab. And then for his postdoc, after having a postdoc in Altus Institute of Biomedical Sciences in Seattle, he find an idea to make it into a company, and we are looking forward to hear his talk about the research which he's doing in his company, the Talus Bio.
Yeah, thanks so
Floor
much for
is
having
yours.
me. This is such a cool community, and I'm excited to be plugged into it going forward. And that's exactly right. A lot of this work does date back to the time in academia at LTS in the Stamatoinopolis lab. I'm from the ENCODE project, and really thinking of ways to look at the gene regulatory system in a new way. But I want to just take a minute or two to go even a little further back and explain a little bit of the motivation behind why we wanted this assay to exist in the first place. My early training was in a chemistry lab. Yeah, I was a chemist by training, and back in the late aughts and early 2010s, the undruggable frontier were some of these proteins that we think about all the time. So, bet bromodomains, chromatin readers, writers, erasers, and in 2010, we published the first molecule to bind and inhibit a BET bromodomain, JQ1. Hopefully, many of you are familiar with this. And at the time, that was really exciting. We saw JQ1 having activity in many different tumors. The data on this slide is showing an actual BRD4 rearranged tumor, actually differentiating from these round, fast growing tumor cells to these more epithelial cells. The molecules work beautifully in vivo. And we saw this over and over again in leukemia, other solid tumors, even non-cancer applications as well. And the reality, the update since then is that as these molecules have moved into the clinic, into phase two, they've had a lot of struggles continuing to move forward, is the reality. And maybe we could have predicted this. BRD4, actually, all of the three targets of these molecules, BRD2, 3, and 4, are all expressed ubiquitously across nearly all of the cells in the human body. So the toxicities that we're seeing in the clinic have been challenging to get over, and we're seeing this more than just with the bromodomains. HDACs, which we do have some improved inhibitors for, still show great toxicity, and that's a big challenge to manage in the clinic. EZH2 inhibitors we were able to get approved recently in certain settings, but it's definitely becoming a theme for some of these chromatin cofactors that, due to their widespread expression, they're challenging to use as drug targets. But we know from things like Project Drive and DepMap and these large consortia that there are good drug targets in the gene regulatory space, but they tend to be transcription factors, tissue restricted master regulators, either of development or growth. A few of them are just shown here, but I think the last talk also illustrated nicely that when these proteins have such high levels of intrinsic disorder, they can be very difficult to study with traditional methods. And we see this despite hundreds of targets, hundreds of transcription factors being validated across many different types of tumors. We have 8 transcription factors with approved drugs, a few more in the clinic, and most of these were drugged in the 50s and 60s. They're mostly hormone receptors or cases where we get lucky and we have a structural crystal structure solved that allow us to engineer a small molecule using traditional tools. So how do we get around this disorder? Are there ways that we can use more modern technologies to try and access these targets in a setting where they're actually full length, folded and functioning properly without having to try and bring them in vitro or tag or manipulate in a way where we disrupt their function? And that was really the inspiration for a lot of the work that we're doing here. How do you build an assay to access these targets so ultimately we can understand how they're behaving and try to manipulate them in a therapeutic way. So this is how we thought about it. These were the kind of the requirements of the assay we wanted to build. This is an assay to measure transcription factor activity and really activity of anything that's interacting and regulating the genome. We wanted to assay these transcription factors in the most relevant setting as possible. So ideally, for us, that would be a cancer cell where these TFs are a driver. We wanted to avoid tags. We wanted to avoid fixation. We wanted to avoid artificial gene loci. So that rules out a lot of the traditional expression luciferase reporter vectors that are being used. And then also we needed it to be scalable. Drug discovery requires testing thousands to hundreds of thousands, sometimes millions of molecules to get the final clinical candidate. So we needed to be able to do this, not just at scale, but in a way that's precise. And the way we thought about this, I was in the ENCODE lab at the time, in the lab that really was pioneering DNA-seq. So we were really familiar with all these different methods, and we wanted to try and figure out how could we kind of adapt some of these ideas and move into something that gets at these requirements. So the most obvious first approach is something like ChIP-seq. So here, ChIP-seq, I'm sure most of the folks here are familiar with this. It's a functional genomics assay, so we're using genomics to measure the function of a protein. Here, we use genomics to measure the amount of transcription factor bound to the genome after usually fixation and pulling that protein down. So it lets us do some of these things, like look at TF activity and looking at their native gene loci, but it doesn't check all the boxes. It does require fixation most often or tagging the proteins. It's certainly not scalable, especially antibody-based approaches. And we only can look at one transcription factor at a time here. So, in drug discovery, we want to be able to see what a compound does more globally to know if we're actually getting a precise effect, or if we're just blowing up chromatin or intercalating DNA and something that's going to be toxic. The other kind of type of assay that we started to learn from are these chromatin accessibility assays. Our lab was really into the DNA-seq. Obviously, ATAC-seq has grown rapidly since then. But some of these key ideas really allowed us to think about this in a new way. The benefit here is that these assays don't require fixation. They don't require TFs being manipulated in a way that removes their native context. The problem was just that we're not actually directly measuring the protein itself. The way these assays typically work is that you're either looking at tags or cuts in the genome where the transcription factor is binding nearby. So we're able to kind of guess which transcription factor it is based on the motif. But because of that, it's not as precise or robust as we would have liked. So what we came up with at the end of the day is kind of taking some of these ideas, but flipping it. Instead of using genomics as our assay to read out the activity of the protein, proteomics had gotten to a level where it became scalable, robust, and actually an option for us to think about here. So we moved towards a functional proteomics assay using proteomics to measure a protein's function. And that's this native Regulome profiling. I'll tell you a bit about that and how we've got it to scale. So this figure is a little unnecessarily complex, but it does capture the idea of what we try and do at Talus. So essentially, the idea here is we can take a cell in its native state. Usually, these are cancer cells growing in a dish, mostly in 96-well plates at this point now. And then we do these sequential steps to purify different fractions of that cell, specifically looking to hone in on the chromatin and understand what's happening there in a dynamical way. So we have these 96-well plates. We actually had to invent these new magnetic beads that allow us to, unlike concanavalin A, which binds not just the nucleus, but lots of other glycoproteins in the cell, beads that allow us to specifically bind the nucleus and fish it out of a gently lysed cytoplasm. That nucleus is then exposed to several different conditions that separate first the unbound proteins floating around in that nucleus, which we call the nucleoplasm, and then from what's left behind, separating that into what we call the chromatin fraction. And actually, it requires two steps. So we have a soluble chromatin fraction of more loosely bound proteins, and I'll show you a little more about that. And then there's also this insoluble chromatin fraction that contains some of the things you might expect: histones, CTCF, some of the very tightly bound proteins. And often what we'll do when we're doing drug screening is combine both of these together into one chromatin fraction. All these proteins are then processed in high throughput in standard bottom-up proteomics. So the proteins are all digested using trypsin, uh, reduced ethylated, and then we use these new time of flight instruments that are very sensitive and robust to quantify in each of these fractions not just which proteins are present, but actually be able to quantify the amount of protein present in each of them and how they shift between fractions over time. So this is just what this looks like. This is one well of data that we're able to get out of this assay. So here, every line is a different protein. And I'm going to show you a few different, these few different fractions in a row. So first, like I said, we take these cells, we do this gentle-lysis, patch that nucleus onto the magnetic beads, and what's left behind is the cytoplasm fraction. And as you can see, or at least in figure, most of the things we call out that are interesting in chromatin are not very highly abundant in the cytoplasm, as we would expect. Same with the nucleoplasm. And then as we go into chromatin, we start to see some of the things that folks on this call are probably very interested in. So in this first data set that we looked at, which was just from one cell type, the cell expressed about 30% of the known transcription factors in the chromatin fraction. We were able to detect 65% of the known cofactors, and then more than half of the RNA binding proteins annotated from these various databases. Interestingly, the RNA binding proteins are also pretty highly enriched in nucleoplasm as well, which wasn't a surprise for us. And you might fairly ask, and we get this question often, is this even useful information? Could we just impute or assume or guess how much protein is bound to the chromatin based on how much protein's in that cell or how much RNA is in that cell? And we try to answer this question or start to, and the answer right now is no. So you might have seen these plots in the past where we compare RNA and protein levels in the cell, and they do correlate. They correlate weakly with R values around 0.4 or 0.5, and that's actually what we see here. So if we compare the amount of protein actively bound to the chromatin to the amount of RNA in the cell, we see a loose correlation, but it's weak. And same if we look at protein levels, maybe slightly higher of a correlation than the RNA levels, but the amount of protein in the cell does not very highly correlate with the amount actively bound to the chromatin at any given time. And I think that's consistent with how we have seen gene regulation studies progress over time and our understanding evolve, where many of these proteins can be sequestered in the cytoplasm, activated by phosphorylation or DUBs or all these different types of regulatory mechanisms that are hard to capture in a global way. So this is the assay at a high level, and I'm happy to share more about how this works. We did put a preprint up about this a few weeks back, and I'll share the link to that as well. But it does check all the boxes for us. We can look at TF activity in a native cell, not just one at a time. We can actually see the activity of all transcription factors simultaneously and all genome-bound proteins simultaneously, for that matter, without having to tag or fix cells. And we can do this at high scale, which I'll tell you a little bit more about how we made that possible. In the preprint, we also were able to put out the first initial map and looking across tissues to see how different transcription factors and proteins are regulated in these different tissue contexts. This is a little bit of how we think about this. These are a few tissues that we care about most at Talus, where our drug programs are. But as you can see, here we see across the entire cell atlas, we see the androgen receptor, which is a known driver of certain types of prostate cancer, very highly abundant in these two cell lines that are AR dependent. TBXT, this is another developmental transcription factor we care a lot about. This is our lead program that'll be in the clinic soon. This is a developmental transcription factor that's normally shut off in any other normal tissue in the body, but is reactivated in these spinal cord sarcomas called chordomas, and we see that very specifically here. And of course, We can see lots of these tissue restricted patterns beyond what I just pointed out. And I encourage you folks to dig into the paper to look a little more deeply at their favorite tissues if that's of interest. We can kind of look at this the other direction as well. Like where do transcription factors fit? Can we actually find transcription factors that are enriched for certain tissues that we didn't realize before? Now that we have this ability to look globally across all these transcription factors, and I realize this is hard to read. We will be updating this for the before the actual publication comes out. But I think some of the patterns here are very interesting and in general. I mean, we have this cluster in that sort of east side of the plot of these common transcription factors that tend to be expressed more broadly. TEAD is one of these that are being pursued in the clinic right now in oncology. MYC, which also is a broad transcription factor, is in this direction. We happen to have a lot of highly MYC expressing lung cancers in our data set, which is why it ends up getting pushed a bit to the north. And as you can see, we have this like nice neuroblastoma cluster up here with the lineage specific neuroblastoma transcription factors, medulloblastoma, a leukemia blood cluster down here, and then sort of a female hormone dependent cluster down here with ovarian cancer and breast cancer. And we've tested about 40 of these cell lines now. We're starting to get closer to saturation, at least from what we can access in the oncology space. The next step for us in building this out is trying to really move to new developmental settings. And if that's something folks are working on, interested in trying out, we'd be very interested in talking more, whether that's uh, stem cell based differentiation pathways or other systems outside of oncology. I think it would be really important to understand those more deeply. One of the other exciting things that this lets us do is this assay is very fast, so we can actually look at dynamics on the order of minutes or hours, which if your immunology is pretty interesting and exciting as well. This is a small vignette from the paper. I won't go into too much detail here, but we looked at a handful of these different immune stimulants in primary human monocytes. And we can see many of them going through the known pathways. So this very canonical LPS stimulant activating the NF-kappa B complex, the STING pathway activating very specifically this IRF3 transcription factor. These are all very well known, but we can start to discover other transcription factors as well that might be playing a role that were not previously appreciated. We actually see this very poorly annotated zinc finger transcription factor, ZFP3, as a commonly activated factor across diverse stimuli. And we're looking more into how that might play a role in the innate immune response. We can also use methods like this to start to add more information and dissect some of the protein complexes we care a lot about. Here, this is an example of a study where we were looking at different modalities and types of inhibition of the menin complex. There are folks working on these irreversible covalent menin inhibitors versus these more traditional reversible menin inhibitors, excuse me, and asking how are these actually different? And we can start to see that when this is just the example of the covalent molecule very strongly disrupts menin itself and one mother member, MLL2 from the menin complex. But most of the complex stays very squarely engaged with chromatin and even a little bit of compensation for some proteins on the other end of the complex, which is a bit different than what we see with the non-covalent inhibitor. Of course, more on that in the paper, but another sort of thing that we're excited about and looking at with this method. So what are we actually doing with it at the company? So I'll wrap up quickly with some of the applications that we're using Regulome profiling for. So now to date, we've looked at enough samples that we have 100 million interactions that we've seen between small molecule drug-like compounds and Regulome proteins. And if we think about how that compares to the scale of other chemical approaches, chemoproteomic approaches in biology, we're about an order of magnitude higher than what's been published, and it's growing very fast. We showed you some of the diversity in cell types. Right now we're measuring, this is a bit out of date, you can do about 2,000 of these assays a week. And this data is getting to the point where we can actually start to combine it with these foundation models in the community and leverage this to start to learn some of the rules, the chemical rules of transcription factors. This traditional undruggable class of proteins is partially undruggable because we don't know what chemistry to try. You can imagine billions of molecules that might be able to bind and inhibit a transcription factor, but since they're disordered, we can't use things like bolts or AlphaFold to try and narrow that space down. But now with this new type of data, we can actually go into those large theoretical compound libraries and at least start have a starting point, have a list of things to try first. And for us, what we're starting to see is that depending on the target, we can predict, synthesize, and test a few hundred to thousands of compounds and actually get hit rates in the 1 to 10%, which may sound low for an undruggable target, where typically the hit rate is 0%. This is very exciting, and these are proving to be good starting points for us therapeutically. One of the molecules we're most excited about is this molecule that actually binds to an RNA regulator. It doesn't inhibit this protein, it just binds and slightly changes its activity. And the activity we care about most is this androgen receptor variant in prostate cancer. I won't go too deep into the biology, other to say that the way we normally treat prostate cancer is by blocking the androgen receptor and blocking its hormone binding domain, which normally binds testosterone. But cancer is smart and it can just chop that domain off, so the drugs no longer work, and the androgen receptor transcription factor can still go ahead and bind to the genome and activate genes. So what we actually were able to see in our data, since we can measure all these proteins simultaneously, are these two simultaneous events happening with a series of molecules, and we can now use this as an engine to move these molecules forward. So we found molecules that can specifically bind and engage no-no in an irreversible way. So what does that mean? The compound binds to no-no, and it changes the mass of the no-no peptide that we observe in such a way that that peptide actually decreases in abundance. We can then look, we do this no-no engagement assay at a short time point, 2 or 4 hours, and then we can look later, 24 hours later, does it actually affect the androgen receptor abundance on chromatin? And we can see that these two measurements correlate very highly. We've gotten molecules now that are very robustly stripping the androgen receptor off of chromatin when we engage no-no. And of course, these two things correlate quite well. And these molecules are moving into in vivo evaluation in the next few weeks. So we're very excited about that. And I want to just wrap it up here. I'll thank the funders for us, which is a mix of venture capital, foundations like the Anti-Health Care Fund, corporations like Bruker, as well as more traditional funders like the NIH, NSF, and the Cordoma Foundation. And last, I'll just mention a few ways if folks are interested in learning more to engage with us. We have a technology access program where we're providing access either to kits or to the assay itself for free for really interesting applications of these types of technologies. We're excited for it to get out there. So either email me or fill out this form. I could send a link out to the group here after the talk if people are interested in that. We have the Regulome Atlas and some of our early small molecule predictions up on this portal, regulo.me. And then if you have other ideas or are interested in collaborating around new molecule discovery or profiling, just send me an email. Be happy to talk more. And with that, I will wrap it up there. Thank you.
Thanks, Alex. That was quite interesting. I know, so I will ask the first question. I know
Please,
you
yeah.
uh company is more going like cancer specific and so, but will you be only focused on human, or are you thinking about other model organisms, or yeah, basically.
Most of our work isn't human. We've had to do some mouse work for
Mhm.
the discovery work, but fundamentally, if there are methods available that can isolate nuclei from the cells of the organism, it should be compatible downstream with with the assay. And then on the analytical side, it's just a
Because
matter
it
of
looks like it, yeah. Exactly. It's
swapping in a
really
different FASTA. Yeah.
Yeah, because I'm coming from the fish field, and the opportunity is endless. So, any question? Somebody has raised their hand. Stephen Krovitz, I will unmute you so you can talk, Stephen. Yeah, Stephen, you can ask your question now.
Okay, listen, excellent presentation, very, very enjoyable. Quick question there. So you had actually fractionated chromatin into an insoluble, soluble, and nucleoplasm. Now,
Mhm.
typically, at least in the old days when that was done, it used to form this structure called the nuclear matrix, which you'd find a center core, which was insoluble, and then these loop domains, and then the nucleoplasm. And typically what you would find is you'd find two things. One is that the loops would actually change the position, so that it would change the position in relation to the structure, the insoluble material, which is quite interesting, depending on what the cell was doing at the time. And then the other thing is that the components of the nuclear plasma would also change. Have you actually gone to that as really the point for the mechanism?
So we have compared these data with these methods from the past and see very high concordance. Often the proteomics done uh, for methods like this is very crude, so we don't actually get to compare many different proteins. But for the ones that we do compare, they are, you can find some of this in, there's actually a paper in 2020, Federation et al. 2020 that sort of presented this method at first. I will say the actual chromatin purification, the nucleoplasm removal happens stepwise. So we isolate these beads, these nuclei onto beads, wash out the nucleoplasm with our specific reagents first, and then the chromatin is purified. So we don't think that the chromatin structural changes will be impacting nucleoplasm, the composition of nucleoplasm, since it's already been extracted. Um, and then yeah, on the looping side, we can't look at that. We can't look at positions of things either. I think that's one of the big downsides here is we lose the spatial dimension on the genome. We're kind of collapsing everything into one measurement per protein, but able to do it over many, many samples.
Thank
Great,
you.
thanks.
Yeah, thanks, Stephen. Thanks, Alex. So the next question in Q&A is from Maria asking, curious, how good is your histone capture with the MSSA? If we have mutations that decrease nucleosome stability, would this be something that can be detected with your method and something you would be interested in? I will reach out if so.
That's a good question for my co-founder. So she did her postdoc with Ben Garcia before he moved when he was at Penn. So they did a lot of this stuff. We definitely see a ton of histones in the assay. The most abundant protein that we see. In terms of looking at histone mutations, I don't think we've done that. But Lindsay would be in a much better position to answer. But yeah, please reach out and we can continue that discussion. I don't have a good answer for you right now.
So Ben also had a question. Ben, yeah.
Yeah, really interesting work. Couple of questions. First, just a technical one, and you may have said this, but how many cells do you need to have this to work?
Yeah, the kind of like standard number for us right now is about 10,000.
Oh, wow.
That's per well. We don't use all the protein we get from that, so we're definitely not limited by cells. It's more a limitation of time. Like, could we move to 384 maybe? But we have
Just
to like
many
move
cells.
fast. We're a startup or whatever. Yeah.
Yeah. And then as I was listening, I was wondering if the kind of what you're seeing bound to chromatin versus nucleoplasm versus off. Like, are you able to make predictions on things that may be binding together and
Yeah,
interaction?
we're starting to get data about that. This I'd encourage, this is just the first few panels from this figure. This is sort of where we started to test some of these hypotheses. The dose response works really well here. If we have small molecule probes, just because it lets us be able to do these correlations overdose and actually model when these different things are coming off. So we can actually see the proximal effects of complex disruption versus potential secondary or downstream effects. Also, time point matters a lot here too. We do these at a really short time point, so make sure that we're not getting transcriptional feedback loops influencing this too.
Mm-hmm. Yeah, this is really cool. And then I was looking at your regulo.me, and the small molecules that you're predicting with AI, are those just like a whole mix of various things, or I may have missed what the kind of underlying theory is behind it.
Yeah, so essentially the way that that's working, this is not the clearest figure, but what this does is that we sort of bring in data from really three sources. We don't even show all three here. One is the data we've collected. So we've now seen, we have 100 million examples of how a small molecule impacts a regulome protein. We actually pull in foundation model embeddings for both chemical space, that were trained by others in the field, as well as protein space. We use ESM for that. And then we also combine this with a theoretical chemical library of things we can make very easily, but still covers this vast, wide domain of chemical space. And then what the model really does is it's not designing new chemistry. It's essentially like a recommender system. So it's taking this 2 billion molecule space and saying, these are the first 1,000 things you should test. They're not all going to work for your target, but we only need one as a starting point. Yeah. So it's definitely a way for recommending and triaging and trying to learn some of the rules for these different protein classes.
And how much are you baking in selectivity? Like if you've got similar paralogs, is this saying this is going to be specific to this protein versus any other, or is it saying this is just going to be a good thing for this protein?
Yeah, so when we do these recommendations, you're right, we do get it's a matrix-based method. We get a matrix of how every compound affects every protein. So we actually do have a selectivity cutoff that we use when we're deciding what to actually make and test.
Yeah, that's cool.
It's all they're all predictions, so most of them are wrong. But it's a nice heuristic and starting point for us.
Yeah, that's great.
So the last question is from Genevieve asking, did you compare primary cells to cancer, for example, in blood cells?
Yeah, yes. If you want to read about some kind of fun, ridiculous primary cancer cell work, we've been doing some experiments on ourselves. You can check that out on our Substack and tell us by a blog. But the more real answer is yes. Some of the work on the immunology, really our new programs are going to be moving into the immunology space. So we're looking at different subsets. This is all done in monocytes, but T and B cell subsets as well as comparing those to leukemias. And the last thing, I guess, I don't know if we can talk about it. Yeah, we're moving beyond blood as well, into more traditional tissues, liver, muscle, as well. So stay tuned. I'm sure those will be in the real paper when it's published.
Looking forward to it. Thanks, Alex. That was a really great talk, and also sending for the two amazing talks which we had today. So we need to wrap up as this is the end of our session today. And join us on October 22nd for talks from Alice and Sungsu, which we will post the poster as soon as we have the titles. And I hope everybody has a good morning, noon, and afternoon and evening, wherever you are. See you soon in 2 weeks. Bye.
Bye. Thank you.
Thank you both. Bye, everyone.